We're going to have your attention. So we have a real special treat today. We get to hear from Dr. Angie Ribera, whom you've already met briefly as you got to look at her zebrafish under the microscope. A little bit of information about Angie. She got her PhD from Columbia University, New York. She joined the physiology faculty in 1990, so she's been on the faculty 27 years. And she has had a whole host of leadership positions on this campus. She has been the director of the neuroscience program in the graduate school. She's been the associate director and still is the associate director of the Center for Neuroscience. She has been the associate director for the medical scientist training program. And in 2013, she became the interim chair of the Department of Physiology and Biophysics. And in 2014, she became the regular chair, or as the rest of us call it, permanent chair. But she prefers regular. So in that time, she became my boss, and she's been great to work with. And she's also a very, very well-established scientist. And you'll get to hear a little bit about the exciting work that she does today.
Well, thank you all for coming on a Friday afternoon. I have some questions for you. It's not a test, but just about how these series are going, because I'll let you in on a dream of mine. Okay. But at the outset, I'll say, besides working with zebrafish, which makes me different from probably everybody else in the department who works with mammals, I consider the nervous system in the context of development. And I didn't start off that way. I started off more like other people in the department, thinking about neuronal activity. And I'm still very interested in that. So on the one hand, I'm interested in how the nervous system can process information so rapidly. But as I studied neurons more and more, I got more interested, equally interested in the question, how do you put this thing together? Okay. And how we are also different, and it can't only be genes that account for all of this. It's the nature nurture story, and so I'm combining both of those interests, I think, and I'll explain to you what I'm doing. So we're going to be talking about development, not so much the adult nervous system, and I'm just going to give an overview, put out some numbers that I think capture how amazing it is that we can put this nervous system together. And then I'll turn to my study itself. I'll be talking about how we acquire our sense of touch and the neurons that are involved in that, and then just summarize everything, because it is a Friday afternoon. Okay, so we'll start off with an overview, and this is a very quantitative department, but I'm going to give you some numbers, and we're going to be talking about them qualitatively. Okay. So some numbers relevant to development, and they're going to be 10 to the 6th, 10 to the 12th, and 10 to the 15th. Any idea what they might be? Start with 10 to the 12th. So that's the number of neurons, cells that we have in our brain. And actually during development, most neurons are generated during embryonic stages, and we generate twice as many as we need. So there's also an awful lot of cell death. Okay, so that number is staggering, but remember, somebody told me it's more than all the stars and all the galaxies and everything. Wow. It's a huge number, okay? All right, well, what about 10 to the 15th? Excuse me? Skin cells. We're going to stay in the nervous system, but that's a good idea, okay? So neurons make connections with other neurons, right? And 10 to the 15th is the number of those connections, which are synapses. So on average, every neuron makes 1,000 synapses. So it talks to 1,000 other cells at the same time. I can't do that, all right? And then this is one that I find really staggering, 10 to the 6th. And the unit is meters. So I don't know if in other talks.
Oh, it's like the length, right?
So the neurons are asymmetric. They're polarized cells. They have that long process, which is the axon, which is what reaches the next cell. And then they have their stubby dendrites, which are the point where information comes in. If you took all the neurons in our brain and put their axons tail to tail to tail to tail, that's how much axon length we have in our brains, 10 to the 6 meters. And that allows us to, I don't know if I don't have the, yeah, I do have the units right. You get to the moon and back, or several times around the Earth's equator. Okay.
Wow.
Okay, so that's a big job to put this together. And when the cells that are going to become the nervous system are first specified, it's less than 1 million. So we go from less than 1 million to 10 to the 12th. Okay. You ever wonder why a pregnant woman is hungry? That's a lot of work. Okay. Okay. And then I want to contrast it with another number. So we're talking about 10 to the 5th, much smaller number than any of the others. Anybody have an idea what that is?
Impulse.
What?
Impulses.
No, now we're going outside of the nervous system. Something in every cell of your body. So for me, one of the most humbling things that we learned in the last century was the number of genes in our genome. And, you know, initially it was 30,000, 20,000, 25,000. That's what this is, the number of genes. So if we think of genes as the blueprint for who we're going to become, with 10 to the 5th, we're making all of this. So I think there's not enough information there, right? And so what I think is so fascinating about development is how do we do so much with so little? Okay? And so what I'm going to put out there is that there must be another way that we get signals and cues and guidance during development. And that's how I combine my interest about fast information processing, fast signaling, and development, because during development, cells are also, neurons are also electrically active, and that activity provides cues. And so there's activity-dependent signals, activity-dependent development, and that is what I'm interested in. And it's really how do we, you know, do so much with so little. Okay. It's a way of, what would you call it, meta-signaling, epi-signaling, getting more. Okay, and so the specific case I'm going to talk to you today about are the neurons that detect touch. Okay. All right, so let's now get to the sense of touch. And we're going to just give some background. When does it appear? I'm going to tell you about the role of touch for the fetus and neonate. And then we'll turn to the zebrafish and which are the neurons that detect touch. Okay, so just some background. Anybody have an idea when you began to be able to detect touch? Okay, so if you ever look at ultrasounds, a fetus when it's about 2 months old is starting to do this, and then at some point opens its mouth, and you know, so there's a sense of touch that's already being acquired. It first appears in the face, and then it'll spread to the limbs, actually to the genitalia, one of the first places where you get a sense of touch, and then throughout the whole organism. So it is the first sense that develops. And the organ is really the skin. Somebody said skin. You said skin, right? So when you think of vision, you think of eyes. Okay, when you think of hearing, you think of ears, right? You think of taste, tongue. Well, for touch, it's the skin. Okay? So it is the first sensory system to develop. Most texts say by 8 weeks. There's some that actually say by 3 weeks. Okay, at birth it's the strongest sense, and it's actually thought that it may help prepare for the growth process and aid the growth process. Fetus will participate. Okay, the face and mouth are most sensitive. And just think about it. Infants love massage. They love to be touched, right? And also, what is a pregnant mother doing, right? Doesn't that calm the fetus? So I think that there's a lot of information that we could be getting from the fetus that cannot. And that is one of the things that I'd like my studies to lead to, is how we can get more information about how things are developing. So, because the fetus is doing a lot. Okay? And if you just think of the neonate, if they don't have contact with their mom, which usually means they're not being touched, there's a lot of abnormal development. I mean, there are these classic cases in Eastern Europe with all the wars that happened there, that infants that were in orphanages, they weren't handled, they weren't hugged, and they developed completely abnormally. So, very important to have the sense and to use it, to be stimulated. All right, so it appears embryonically, and that is why I love the zebrafish. Okay, so these, about that one that you were looking at is 6 days old. Okay. And yes, this up here is like a 10-minute embryo. The cell, it's this clear thing on the very top, that's the embryo. And the big ball down here is the yolk sac. And that's actually, you can think of it like a very big lunch bag. It'll sustain the embryo and larva for 5 days. And so you can actually have zebrafish develop that are messed up without a heart, but they'll be okay because they have that yolk. And so it's been a system where we've been introducing mutations into, and so you can uncover things that do a lot of damage, but they make it out pretty far, and you can see the consequence of it. In mammals, sometimes you'll have a gene that's pretty essential, and then it just dies. You don't learn anything from that. So here you actually get it to survive a bit and see what the role is. Okay, so the other thing about zebrafish is that all of embryonic development occurs externally, and this is true for many species of fish and frog. The mom deposits eggs, and the male comes along and fertilizes them. So this is all occurring externally. So we have this easy access to the embryo. And per mating, you put a male and a female and they're happy, you can get at least 200, sometimes 1,000 embryos, compared to the litters of mice, which are maybe 4 to 6. As you saw, it's a transparent embryo. But think about it, if you were interested in heart development, that heart begins to appear at 1 day. You could look at the same embryo at 1 day, 2 day, 3 day, 4 day, and so you're looking at the same specimen, and that gives you a lot of power. The neurons that I'll be talking to you about, they develop early. They're present before 1 day, some of them. Okay, we'll get into that. And they'll elicit some behaviors. Okay, so you can look at it at the level of behavior. And then it's a system that has become very, that's a phone. Okay, I thought it was a car driving by with, hard rock or whatever. So, but it's amenable to genetics. So we can introduce mutations randomly. And now with CRISPRs, which I think you all have heard about, we can actually target genes for mutations. And then transgenics, we can introduce engineered genes. And what's nice about this is we can introduce genes where we'll have one element that says, oh, you're going to be expressed in this cell, that cell, and that cell. And what is being expressed is a fluorescent protein. So you can actually very easily see those cells. And we're going to see examples of that. Okay, so lots of genetics, mutants and genetics. Okay, so we're going to look at some behaviors now. Okay, so this is a 2-day embryo, right? And that's the touch response. Okay, that one that was there didn't have it because I put some tricate in the bath and it was anesthetized, right? And so it's a pretty unambiguous behavior. And so if you want to screen for touch sensitivity, this is a really nice assay. Okay. What are you poking at? Oh, the hyper, a little poker.
Tip of like a pipette.
But you can't hurt it. Like it's so
Oh,
small.
you
It
can.
seems like
One of these, you can actually see the tail is a little bent, the one on the top.
Oh.
Yeah, I got a little enthusiastic. These are not responders. Okay, so actually, this is the first. I started my work with zebrafish when I was on sabbatical, and this is a mutant, and it's not dead. The heart is beating there.
Oh, yeah.
Okay, so maybe it wasn't responding because I, paralyzed it. But no, it's a mutant, and it's not moving. Okay.
Interesting.
And that's what got me into the model. Okay, but I just want to show you that this is a very efficient assay to uncover mutations that affect touch sensitivity. You can watch these embryos and see that they'll swim spontaneously, so you know it's not a problem with the muscle. They can move, they just don't move when they're touched. Okay?
Oh, because I thought all fish had to keep swimming or they die.
No,
Oh.
they sleep.
Oh, okay. All right.
Okay.
Yeah.
Yeah.
If they can move their gills and keep the
Yeah.
oxygen. Okay. Yeah.
Just like us.
Yeah.
Yeah.
Okay.
Okay.
I
Yeah.
didn't know they slept.
Oh yeah. Yeah. It's becoming a really powerful model for study of sleep behavior and the neural mechanisms that regulate sleep. Yeah.
Interesting.
How long do they live?
The zebrafish, they live for a few years.
Really?
So, let me go back to this.
Oh my
All right,
God.
so here, thank you. Okay, so here, this is just like a 10-minute, and down here, this is 1 day.
Okay.
Okay, so these are different stages. You can see that, a few divisions have gone on. You can see a few cells here, a few more. And by 1 day, it already has an eye, a heart. It can move.
Mm-hmm.
Okay, so it's rapid development. This is 2 day. This is the stage at which I was doing the touch assay.
Mm-hmm.
And the one at the bottom is 5 days, just a day younger than that. Okay, so I'd say these are adolescent. I wouldn't say they're adult. They're not yet sexually mature. And that happens in about, depending on how much you feed them, you can get them sexually mature by 6 years. So it's a rapid development as well. Okay, so we're going to talk now about touch-sensitive neurons. And zebrafish have 2 types of touch-sensitive neurons. I will make the case that we do also. Okay, and so you see the arrows at the top and the asterisk. Okay, so what's green here? This is an example of a transgenic line. It has the regulatory elements of a gene known as neurogenin, okay, which drives expression of genes in neurons. Okay, and so you can see that there's some neurons, these up here, you can see a few down in here. They're in the spinal cord. Okay, so anything that's in the spinal cord or brain, we call that a central neuron. And then these big guys, they're actually not in the spinal cord. The spinal cord like this, they're out here. And so they're a peripheral neuron. And all of our sensory neurons, for the most part, are in the periphery. So what are the arrows? Those are a cell type called Rohan beard cell. Okay. And they're in the spine. They have a cell body in the spinal cord, which I just told you that sensory neurons are peripheral. So this is an unusual one. It has a cell body in the central location. They're born early, so they are present in the embryo by 13, 14 hours after fertilization. And the dogma is, and we're going to challenge this, is that they're transient, that they're born early and they survive for a limited amount of time and then they go away. And there are a few other kinds of neurons that are transient. For example, in our cerebral cortex, you may know that there are layers in the cortex, and that's important because neuron in this layer has to communicate with neuron in that layer, and if they're not in the right layers, they don't communicate correctly. Well, there's a transient neuron known as a Cajal-Retzius cell that is absolutely essential for telling neurons which layer to go to during development. So even though it's transient, doesn't mean it's not important. All right, so what about the asterisks? Those are neurons they're in the periphery, they're known as a dorsal root ganglion neuron. They're born later and they persist. So they are present in adults. We have dorsal root ganglion. And most of the time when people talk about our sensory system, they're talking about dorsal root ganglion. Okay, and then I love this picture. Okay, here, these are 2 Rohan beard cells. And what we've done here is we've looked at it in the microscope in sections and then collapsed all the sections into one image. And what you're seeing, all of these little tiny dots, are the processes that innervate the skin. So this is a neuron that innervates the skin, remember that sensory organ. So when you touch the skin, you activate these neurons. And the dorsal root ganglion do the same thing. Okay. So that's what's cool about these transgenic lines. With the fluorescence, you can isolate these cells and see their anatomy really beautifully. Okay, and just as a little aside, I often get criticized, why are you studying Rohan beard cells? They don't exist in mammals, they don't exist in humans. Well, this is a paper from 1944, human fetus. Okay, you see that asterisk that's there?
Mm-hmm.
What I'm pointing out is this cell here. Okay, so cells, central neurons that would be in this part of the spinal cord, which is closest to the back, which is called the dorsal side. They don't have, their axons don't go outside. the Rohan beard cell has to have an axon that goes outside because it has to innervate the skin. And so what Youngstrom found are these neurons that have axons that leave the spinal cord. And here it is in higher power. And then this is work that Christian Artiger and Tom Finger did. This isn't a mouse. What they did is they put a dye in the skin that gets taken up by axons and then travels to the cell body. So any cell that's labeled means that it had an axon that innervated the skin. So you can see here there are a bunch of dorsal ganglion neurons that are labeled. You see this one in the spinal cord, looks an awful lot like that one.
Mm-hmm.
Okay, so these neurons exist in mammals. And I think when people say they don't exist, it's because if they are present in large numbers embryonically, it's really hard to have access to the proper stages in mammals for study. Whereas in fish and frog, you can study them a lot. And one other tidbit, the very first neuroscientist who studied Rohan beard cells was Sigmund Freud, written in 1877. But then I guess he got interested in other things. Okay.
His ego took over.
Yeah, he couldn't call them ego cells. All right, so what we're going to talk about now is why are there both Rohan beard cells and dorsal root ganglion? They have the same function, so they're functionally redundant. And the dogma is that they exist at different times and they coexist minimally. Okay, so that Rohan beard cells die when the dorsal root ganglion are present. If that were so, you might expect that if you have more or less dorsal root ganglion neurons, you would affect the time course of Rohan beard cell death. Okay. And people have looked at this in mutants where you can have different number of dorsal root ganglion neurons. It has absolutely no effect. And so that doesn't seem to be the case. And then, you know, if the dogma is correct, you'd also expect that there'd be minimal time when the two coexist, right? That the Rohan beard cells would really get out of the way very fast. Okay, well, there are some transgenic lines, like I just showed you, where you can very easily see the Rohan beard cells. And this was an initial hint that this dogma needs to be re-examined. Okay, so this is now, we're looking on the back of a zebrafish embryo. And these are all Rohan beard cells. And one way you can also tell is, all right, I'll just do it this way. You see their axons are leaving the spinal cord, right? Okay, so they're there. At 4 days, the dorsal root ganglion are present. We still see an awful lot. They're less, okay, but they're also there at 8 days. And by this point, they're supposed to be largely gone, and there are a lot there. Okay. Well, one problem with this kind of way of looking at it is that, you know, we're looking at the same embryo at different times, and I don't know if the cells that are here are really the same cells that were there. Maybe some cells are born later, a different cell type. Okay? So Christina Williams in the lab did the following experiment. What she did is in that a transgenic line where Rohan beard cells were labeled, she injected a DNA construct that would label a few Rohan beard cells with a different color fluorescent protein. It's that you could follow one over time. And this is just a cartoon. You see the transgenic would have a lot of green, and then you have a few that were also labeled in red, and so red and green. Right? So here is an example at 1 day. You see the Rohan beard cells there. And whoops, I went too fast. Okay, so they're there at 3, 6, they persist, right? And they're there until 2 weeks, which is way, and the keeping them in the lab, we're not allowed to keep them beyond this point. But our studies ended that. Okay, so this is the graph of all these data, and you see that, you know, 40% are present when they're supposed to be gone. And so there are a large number of them that are existing to stages when they shouldn't be. A considerable fraction does die early, but a large number persist. Okay? Why? Whenever with development, whenever I see something and you make an observation, it's why and how. Right? Okay. So I'm going to now turn to the dorsal root ganglion cells. Okay, so I had initially posed that the dorsal root ganglion cells instruct the Rohan beard cells to die, and that didn't seem to be the case. Well, maybe it's the other way around. Maybe the Rohan beard cells are training the dorsal root ganglion cells. Maybe the Rohan beard cells give signals to the dorsal root ganglion. Okay. And so we got some clues about this serendipitously. Okay. We were doing other experiments and we noticed some things, and then it was like, oh, maybe we should study that, okay, which is, I think, often the way science goes. Mm-hmm. Okay, so this is work from a lab in Ohio, Paul Henion's lab. And now we've sliced through the spinal cord. This would be the backside, the belly would be down here. These are two dorsal root ganglion, and that would be what we would consider their normal position. And even, this other lab, you can occasionally see this dorsal root ganglion that has this ectopic position. And neurons, to function, they have to be in the right location. So if a neuron's not in the right location, it's not good. So it's a little curiosity, and it happens in in control wild type embryos. Okay? And so this is work from Melissa Wright. She noticed, okay, this is our accident, that when we inactivated a sodium channel, which is a type of ion channel that allows a cell to be electrically active, we inactivated one that's expressed in Rohan beard cells. We found that there was an increase in these ectopic cells. Okay, so these, in the enlargement here, you can see that they're at the right location. Okay, and then you see there are a bunch here that are in that ventral ectopic location. And so by silencing the Rohan beard cells, we got an increase in these ectopic DRG. And so I thought, gee, that gives us entree into studying what is going on. Okay. So Rohan beard cell activity regulated the number that were at this abnormal position. So one question, and we've studied this a lot, you'd say, well, what's happening to them? Okay. And in this picture, there are two colors, okay? And the red and green now mean something else. Red is actually what the green was before, okay? So these are Rohan beard cells up here and the dorsal root ganglion there, all right? And then green are peripheral neurons. Okay, so what we would have here are something known as sympathetic neurons, and except for dorsal root ganglion neurons, they don't label it the green. And these are enteric neurons. You may know that you have lots of neurons in your gut, okay? And that's what's there. Okay, and what I want to point out is this one here is yellow, okay? So this one, it's not clear. We don't have the earlier image, but this cell had started off up here, and it moved down here. And as it was moving, it started to express the green protein. The green protein says it is associated with another identity, which is of the sympathetic ganglion. And so what this is saying is that those dorsal root ganglion cells that are switching their position are also switching their identity. What I find interesting about this is that sympathetic neurons are ones that help us deal with stress. These neurons are changing fate from sensory to sympathetic under conditions when they're not being touched. Kind of stressful, right? Anyway, well, okay, and then this is again in a little higher power. Okay, so we have some normally positioned dorsal root ganglion, and you see this one has migrated and it's become yellow. Okay, so
they can switch their
yeah. So for me, this is, so it's part of the study, but if I take it out of this study, until this point, I thought, oh, gee, neurons are unlike humans. They grow up and they have that identity, and they can never reinvent themselves. We can, but they can't, okay? And here's an example where a neuron is reinventing itself. And when you think about how we have lots of conditions that are neurodegenerative, and the common strategy is to transplant cells, either stem cells or fetal cells, whatever. This is another avenue. Maybe you can coax cells that are in the individual to adopt a new faith and replace the ones that are dying. And then you avoid a lot of other problems. You create new ones, but you avoid a lot of others. So it's a little bit about how we know that the nervous system is plastic and that it can adapt and change. Otherwise, we wouldn't learn. But the neurons themselves, their identity is plastic. Okay, so they become another neuron. So now back. So I said that if we silenced the Rohan beard cells, and I did that by an artificial way by inactivating the sodium channel. And remember, those ectopic ones are sometimes present in wild type and control embryos. So this happens normally. So how do you normally regulate Rohan beard activity? It's by tactile stimulation, the touch. It's a sensory experience. And so this is an experiment that Chris Nopel did, and it's the wild idea that maybe we could regulate the number of those ectopics by growing up the embryos under conditions where they get more tactile stimulation or less. Okay, so this is the control, and you see two ectopics. So they're there. We can just manipulate their number by these. And then when we increase stimulation, there's less. Okay. And then when we decrease stimulation, there's a lot more. All right. So this suggests that, yeah, it is sensory experience that regulates Rohan beard cell activity, and then that somehow is influencing the number of these ectopic DRG neurons. All right. So at this point, this is when another, who's now a doctor, right, an MD, PhD, when she was a student in the lab, we came up with this model. Is that the green are the Rohan beard cells, and the red now are the dorsally ganglion. And both of them, remember I showed you that image of the processes innervating the skin? Okay, so both of them have processes that innervate the skin. Melissa also showed that they overlap. They physically touch each other. And so the model is that maybe when Rohan beard cells are active in an activity-dependent manner, they secrete a factor, which these stars are supposed to stimulate, that talks to the DRGs and tells them, oh, there's sensory stimulation in this environment, keep being a sensory neuron. Which would mean, though, if the Rohan beard cells were inactivated, that signal would not be released, the DRG would not get that, and they then would move. Okay? So that's our model. And so Melissa began to test it, and we thought, what is a good candidate for those kind of molecules that could be released in an activity-dependent manner? And there's a class of molecules that do that, and they're called neurotrophins. And I've listed the three main ones: NGF for nerve growth factor, BDNF for brain-derived neurotrophic factor, NT3 for neurotrophin 3. Okay. So that's the family of neurotrophins, and neurons secrete these in an activity-dependent manner. So what Melissa did is that what she had embryos where the sodium channel was inactivated, and then treated them with NGF, BDNF, or NT3, and to see if that could rescue the effect. And she could, but only with BDNF. Okay. So I mean, the other two are control, right? And Chris did the following experiment. So these panels are the ones you saw before. Okay. And so if we now take the decreased stimulation and add BDNF, you see now we get the normal number. So we're rescuing the effect. Okay, so here's another. So normally we have some ectopics. If you increase stimulation to more BDNF being released, you have less of them. If you decrease stimulation, less BDNF, you have more of them. And then if you have less stimulation, but supplement with BDNF, you rescue that. So this means, so we've added exogenous BDNF. So it says that it can do it. It doesn't tell us that's what's actually going on, right? Okay, so it can rescue, but is it what normally does it? Is it the endogenous activity-dependent factor? And so this is the point I'm going to leave you with. So somehow we have to manipulate the endogenous BDNF, and we're doing that with gene editing. CRISPRs, and Macy Walker has been making BDNF mutants, and then BDNF has a receptor, TRAC-B. Mine has some TRAC-B mutants up there. And so this is our way of testing this model, and it's holding up. So stay tuned. Next time I'll tell you about that. But I'll leave you with these thoughts. Okay, first, to me, I wasn't looking for this, and it totally wowed me. Neurons can transdifferentiate, reinvent themselves. And as I mentioned, maybe this is a new strategy for treating neurodegenerative conditions. And we go back to how I introduced the subject. Okay, so neuronal activity is providing another signal here. It's another telling neurons how to grow up. And so that's how we do more with so little. And it also speaks to the nature versus nurture.
Mm-hmm.
And we can't do very much with the genes that we have. We can't do anything about that, but we certainly can do a lot with how we're raised. Right? And so for me, that gives us great hope. And that's it. Let's take good care of yourselves on the weekend, right?
Any questions for Angie?
What is CRISPR?
Excuse me?
What is a CRISPR?
Oh, okay. Oh, I'm sorry. Okay. So they've been in the news a lot, and it's a way. A CRISPR is actually an enzyme that will introduce breaks into DNA. Okay. And they were first a scientist who studied these a lot is Jennifer Doudna, and she's at Berkeley. She actually was a dean speaker here not too long ago. And they were actually first discovered because it's in bacteria. Mm-hmm. And they're a way that bacteria protect themselves from viruses. They'll recognize the virus is foreign, and then they'll chop up the viral DNA. So Jennifer Doudna and Charpentier in Europe, they studied this. They isolated that, and they saw that there's other components that help that enzyme, the Cas9 actually, target the viral DNA, and they then modified that and adapted it. So instead of targeting it to viral RNA, you could target a gene in a bacteria or in an animal, in a Drosophila, and then in mice. Okay, and so they really understood the mechanism. But it was really by studying bacteria and their immune systems that we learned about CRISPR-Cas9. Now, there have been, just this summer, there have been several reports that have been on NPR, I think even in Time and Newsweek, for human embryos. There have been genes that are mutated that would lead to a disease that in the lab, they've actually corrected that mutation. And so it's powerful. It's scary. It's brave new world. And so to me, it's really, we're using it in the lab, and I think it's really cool and powerful, allows us to do things, but there is so much controversy and debate about their application to humans. Yeah, but that's another example of, when you're not looking for something in science, you usually find something good, and it's serendipity. So at the outset, I said I was going to ask you guys a question, right? So I think many of you have been to a few of these talks, and a dream of mine actually is at this campus is to have a series of public lectures, pretty much like this, for the community. I think Denver metro area population is incredibly curious, and I think they would find, I personally, I'm a geek, so, but I also think it's important because in this day and age, this is basic science. This is not disease, right? Of course, we want to understand the human condition, but this is basic science where you get knowledge that ultimately allows you to treat disease. And so with all of things that are happening politically, I also feel like it's important to advocate for basic science. So just any feedback you can give about how you think these lectures, these series would go over with the general community, with the public, would be really appreciated.
I think it'd be wowed by it. I think that if you could always be paralleling it with what the translational possibilities might be, so that there's something that they can hang on to.
Yeah.
Like say, oh, this could be used for X. So to constantly be kind of paralleling what the implications could be.
I have to say, I did it a little bit here, talking about it. And as a scientist, I squirm when I do that.
Yeah.
We want so much proof before we offer that. Yeah, and so it's a little bit about, we also have to educate basic scientists about how to talk and explain why that, because that is part of the reason I'm interested in this, but I don't, I can't really say that. I don't have the data to say that, so, and it's, being a little bit, we can say things like that with the appropriate caveats and cautions. So.
Okay. I think it's a brilliant idea, Angie, because I think the more people can learn, the more they have understanding. And I mean, as a young person in elementary school, middle school, or even high school, I never saw this picture of basic science ever. And maybe I would have changed tracks,
Well, and just, I have the zebra fish. This is like show and tell.
Yeah.
But it's super cool.
Yeah.
Oh my gosh. Right?
Yeah. So I think it would be great for society.
I also think, I think this campus is fascinating and really impressive, but it's public transportation is helping us, but it is a little bit out of the way. And if there could be just some teasers that get the public here, I think it would help us in many ways. And so I'm wondering, is this something that would get the public? Yeah, that's a good point.
I actually think there are more people with different departments here. I find it fascinating. But they're not always help people.
Oh, yeah.
Yeah, I think we're the only department that does this. That, yeah. Yeah.
And for us, it also, it's interesting to be that.
We started.
And so now it kind of gives us perspective but I don't really tell people.
Frog muscle. Yeah.
We started it because you're our colleagues, and it's sort of like in 2 seconds it's really hard to tell you what we do, and so we thought that to have some sessions where each one of us spend a little time with you. It's a way to get to know each other better, and then we understand what you do better, you do better. Yeah.
Right.
Yeah. Well, this is what it's all about, what we're all working for. Understand life better.
One of our faculty members proposed offering an administration for physiologist class, but I think there'd be a much less interest in that. It's not nearly as interesting as what you do.
I don't think so. I think, as I've been in this role of, I've had other administrative positions, but chair position, the amount of administration I've had to master, oh my gosh. And then I realized, like, okay, yeah, but then if you do this, you can get that. And so it's cool.
Angie, how did you start in science?
So both my parents are MDs. Okay, so I was expected to be an MD, and I rebelled.
I like it.
No, I would always hear about biology. I was set up to like biology, right? I didn't. I'm not that apple that goes far from the tree. I am the apple that falls close to the tree. But but that is how I got in. And initially, I was interested more in genetics, and then I got to college, and I took a neuroscience course my sophomore year. Absolutely brilliant professor, and that changed me forever. And although I have to say, I'm learning more about immunology these days, and I wonder if I had learned about immunology then, well, I probably wouldn't have. I would have just been totally confused about what to do. Yeah, it's all really interesting. And in the end, I'm using genetics.
Right.
So,
And so immunology is the immune system. Is that what it is?
Yeah.
And so isn't your immune system made up of your neurons and your cells?
so immune cells come from the blood, but there's a lot of interaction, and this is a really hot subject right now, between the immune system and the nervous system, neuroimmune interactions, and it's a hotbed, and they influence each other.
So your cells all come from the spinal cord?
So the Rohan beard cells are in the spinal cord. I didn't tell you where the dorsal root ganglion neurons come from. They come from a different structure. It's transient. It's called barrel crest. It gives rise to all the neurons in the periphery. It also gives rise to a lot of muscle and connective tissue. It's the poster child for a cell type that's pluripotent. So people who study stem cells and want to study how you can coax a cell from X or Y or Z, barrel crest cells are really hot. Okay. And the cells, they start off on top of the spinal cord and they migrate throughout the body. They're really interesting. Christian Ardinger studies them a lot. They're really interesting. And when they are messed up, you get a lot of craniofacial defects, cardiac defects, sensory defects, gut defects.
Interesting.
Yeah, like Hirschsprung disease. I don't know if you know about that. That's people have a lot of enteric problems, stomach problems, and it has to do with the enteric nerve.
I think these are really important talks and could be extremely beneficial because I think people take for granted. So like I go to the mechanic and he's like, oh yeah, you need this. And I'm like, yeah, because I don't know. And it's the same way sometimes when you go to the doctor, like you don't know. There's whole worlds going on. And I think that's why these are so cool, is because you get to hear about like you don't really think about like your cells firing that way when you touch a hot stove. And then you're like, oh, there's things happening. There's like millions of things happening just to make my hands talk because I'm Italian, you know, so.
And a lot of these things are entering politics, right?
Right.
CRISPRs,
Yeah.
that controversy, using stem cells, fetal cells to treat disease.
Right.
Okay? I think the public, because it's so easy
Right.
to spin a crazy story.
Right, right.
So I just think that it's really important and just life is so much better when you're curious.
Yeah.
So,
I think one thing these talks have done for me is it's easy just to work here and think we're doing our job and that's it. But what's really going on here is cutting edge science. Each person we've heard talk, and what they're on the verge of either things they just discovered or are about to discover, and they're it's stuff that nobody else in the world is doing, or a very, very small handful of people in the world. It's absolutely fascinating to be part of something so much bigger and exciting than putting together a budget or routing a form of OGCs. As much as we love to do that.
no, I think I have a great job and I work with lots of great people. So, yeah, it's a classic example here, being a very diverse community, and it works.
Yeah.
Sometimes I'm surprised that the science can actually carry on given all the
Yeah.
things that the stumbling blocks that you have to go through administratively and bureaucratically and getting funding and yet it does here and you still have enthusiasm. Everybody here is so it's
Yeah, I mean, it is hard. And, to some extent, you have to be a masochist because you get so much rejection. But, yeah,
Okay, guys.
Well, thank you,
Yeah,
Angie.
thank you for coming, and
Yeah.
I wish you a really nice weekend. Take good care of your skin.
Yeah.
And we'll be meeting next month, and we'll hear from Dr. Kathy Thorne, she studies pacemaker cells in the heart. We're going to have to reschedule it because we have a conflict. Actually,